So welcome all for this second broadcast of this interesting and intriguing chapter in Gem Pharmatech's journey. And we're going to present some new models today to evaluate drug development for NASH. We're going to give um, others a chance to enter the chat room and probably start in a couple of minutes' time. So please be patient. Thank you. All right, everyone. I think that other attendees are slowly coming in. My name's Pradeep. I'm VP of Business Development here in Europe. I'm based in London, and it's just dinner time over here. And I had a good meal, but today we're having a three-course meal. We're having, for starters, we're having Dr. Mark Moore introducing Gem Pharma Tech. Then we're going to have the main course by Dr. Zhiying Li, who's going to talk about the NASH models. And Dr. Lee's actually surprised me, saying that we're going to have some dessert as well. And we're going to have, well, she's going to go on to talk about liver models as well. So that's exciting. So I'm going to introduce both of them, and then Mark will take over and lead on to Zhiying. So who's Mark? Who doesn't know Dr. Mark Moore? He's a legend in the genetic engineering mouse field. If you don't know him, I don't know why you don't, but if you don't know him, he did his undergraduate in biochemistry at Princeton University. Then he went to Brandeis University to do his PhD in molecular biology. Then he was a senior scientist at Genentech. And after that, he embarked on an entrepreneurial journey where he was CEO and co-founder of the famous Deltagen, which was a very successful company. And due to his successes, he was appointed for a major role in an NIH initiative called the Knockout Mouse Project. Then he served as executive director for the International Mouse Phenotyping Consortium, which is known as IMPC. So two major roles there. And he excitingly joined Gem Pharmatech last month in January. And before that, he was CEO of Life Edit and also Glycologic. So that's Dr. Mark Moores. He'll be talking to us about Gem Pharmatech US. And then, lucky for all of us, we have a next superstar colleague who's going to talk about NASH models. Dr. Lee actually did an undergraduate in Beijing University in life science and biotechnology. And then she got an amazing opportunity to do a PhD in molecular and cell biology at Harvard. And she researched on synaptic transmission and vesicle recycling, which sounds complicated enough. And then she tried to make it even more complicated because that's how clever she was. And she went on to do a postdoc in a different field in metabolism and learned a lot of expertise in metabolism. Hence, she's going to present the NASH model here today. And her postdoc was concentrated on how different neuronal cell types participate in leptin signaling and energy homeostasis. So she's got a lot of experience in that kind of field. And then after her postdoc, she went and worked for Taconic for 3 years in the genetic science and compliance team. As I told you, Dr. Lee is going to talk about NASH models and also end with some cool, interesting data from us on liver models as well. So first, it's Mark and Dr. Moore. Please take it away and introduce us to Gem Pharmatech US.
Thank you, Pradeep. It's a pleasure to be here today and to meet the people online and give you an introduction to Gem Pharmatech, but also to take you back a little bit in time and tell you a little bit about the history of genetically engineered models and the vision that we have for that in the future. So as many of you are aware, mouse research and animal research has been a critical part of physiology and medicine for over 100 years. But in fact, it's been involved in nearly all of the Nobel Prizes. So approximately 186 of all the recipients in physiology and medicine utilize animal models in their research. And the use of animal models really accelerated dramatically with the combination of classical genetics and molecular biology burgeoning in the late 70s and early 1980s. And in fact, as molecular biology was developing, it didn't take long for people to try to implant genes of interest, and there were not very many of them in 1982, into the mouse genome. And Ralph Brenster and Richard Palmer were the first two to do this with a nice Nature publication and launched an entire new field. It wasn't that much longer to the technology improved and the tools improved to where we could then manipulate embryonic stem cells. And this work was pioneered by the Nobel Prize winners listed above in 2007, who combined the molecular biology of homologous recombination with the pluripotency of embryonic stem cells, where you could manipulate in a tissue culture dish ES cells, screen for the genetic modification that you wish, inject it into a blastocyst, and end up with a chimeric mouse, which is composed of both the cultured ES cell and the host blastocyst. This can then be bred out to derive your heterozygous mice carrying the genetic material. This was a tremendous tool. I looked at this when I was a postdoc and said, this is really hard. How many other people are going to do this? So I think I will do it. And the answer was everybody would do it. And in fact, so many people were doing it that when I started working with NIH as a consultant, we did a research project and found that the average knockout mouse publication was created 6 times. The world record was 36 different labs for one oncogene. So with seeing that, there was a tremendous waste not only of financial resources, but time delays in getting the animals into the hands of people to do their work, and also the notion that, again, this is a waste of animals, and the notion that we would like to reduce the use of mice wherever possible. So in a project that was led by Francis Collins, who at that time was the director of the Human Genome Project, he assembled a team of people from all over the world, not just the US, to discuss what shall we do with the mouse genome. At that time, it was predicted there were about 25,000 mouse genes, and there was reasonable information on only about 3,000. So what do you do? Do you do 1 million enzyme assays on unknown genes? And the notion really emerged that we should be doing reverse genetics. Knock out the gene, analyze the mice, and see what's happening by subjecting them to phenotypic analysis. This was being done in individual labs. It was being done in relatively high throughput with companies like my company, Deltagen, and the competing company called Lexicon Genetics, founded by Alan Bradley, one of the pioneers in ES cell technology. And again, we were seeing that this was developing a rich amount of data on understanding gene function, and we wanted to make this available to everyone. So the NIH funded, and also the European groups funded, the effort to create a knockout for every protein encoding gene in the mouse genome in ES cells and make them available to the public. This was very successful. People didn't think we could do it, but within probably 3.5 years, we had nearly completed the mouse genome. So the next idea was to take it further, to move up that pyramid you saw in the past and actually turn these ES cells into mice, and then do comprehensive phenotyping. Again, moving further down the pipeline, reduce the redundant effort of others, and also provide more information to the scientific community. So I was honored to be elected the first executive director of the IMPC, and I helped build a team of 16 different institutes in 13 countries internationally to go after this entire collection of genes and creating mice. This was going along very smoothly in 2010 to 2012. And then in 2012, everything changed. The CRISPR technology came out, the publications were again produced, and if we thought there was a frenzy over ES cell technology, it was nothing compared to the CRISPR craze. As you can see the quote here, there's never an example of any field moving faster, and CRISPR was being used for everything. And again, this was not lost on mouse geneticists, that if you could then simply design a guide RNA with a sequence of interest, you could go in and cleave DNA, which could stimulate homologous recombination, or if you design 2 of these, you could cleave out a gene of interest or a section of interest. And in fact, the IMPC moved on this technology very quickly, despite the success of the ES cells, which you see above. in creating mice. We found that with the CRISPR-Cas9 technology, we could cut that in half. But in fact, it was even better because we cut out all of the ES cell work, which is very expensive. Sometimes the ES cells, you could not get homologous recombination, or you might get a frequency of 1%. So with CRISPR-Cas9, the efficiency is now approaching 100%. So not only does it cut the time in half, it goes from about a 50% success rate to nearly 100%. With this also, we're now able to do much more, because of the efficiency, much more sophisticated genetic manipulations than we could in ES cells. Not just homologous recombination, but amino acid substitutions and humanization of genes. So in parallel with what was happening with the IMPC, and a member of the IMPC was led by Dr. Gao Zhang, who founded the MART, which is a Model Animal Research Center in Nanjing in 2002. They very quickly developed a resource in China that became part of the Asian Mouse Mutant Resource Center and Consortium. And they also became a national laboratory and an animal center in 2010. I met Dr. Gao about that time and recruited him to actually join the IMPC so they could use their technology, depth of science, and also their capacity to contribute to this international consortium. And over the years, they contributed thousands of different mouse lines. About 2017, though, Dr. Gao realized that he had been serving a great deal of the scientific community from an academic setting, and it was time to move this to more of an industrial setting to, again, provide additional platforms, more technology, and other services that really were not appropriate in the academic setting, has received funding from blue chip capital companies, and has now opened 3 different subsidiaries in China, and more, I believe, are in the works. I'm very happy to say that I'm here today because one of the expansions that Jim Pharmatech has done, and this began really in earnest in last month in January, and we just had a press release announcing our expansion and opening of facilities in Massachusetts and in California so that we can bring many of the resources that have already been created at Jim Pharmatech, from humanized mouse models to knock-ins, knockouts, to conditional knockouts, to customers, both in academia and in industry in the United States. And as you've already met our colleague, Pradeep, also into the UK and Europe. So just to give you an idea of what Jim Pharmatech is, I've mentioned a lot of the numbers. Picture's worth 1,000 words. This is a state-of-the-art animal facility. As you can see, a massive microinjection suite with the capability of creating over 5,000 new genetic models per year. That's really a staggering number. Again, that's something that the IMPC was doing about every 5 years. So the capacity and throughput is tremendous. It's ALAC accredited for over 13 years now and upholds the highest international standards, both in animal husbandry as well as looking at microbial references and the SPF status of the animals. So just as an overview, and I won't take too much of your time, please visit our website. The Jim Pharmatech group has really become a one-stop solution for mouse models. As I mentioned already, from standard and conditional knockouts, humanized mice, immunodeficient mice, germ-free mice, all of these are available and you can order them. We'll also do customized services. We are extremely efficient with the CRISPR-Cas9 technology. As I said, nearly a 100% success rate in doing point mutations, knock-ins, knockouts, as well as other transgenic services, what we call super transgenic. So be happy to tell you more about that at another time. We also offer potential customers, again, the one-stop shop. We'll do customized breeding, cryopreserved lines, expand them, rederive them, and ship mice to you for your own research. But also, if you are not an expert in an area or you don't have the capacity to do it, or you want to try additional experiments, we'll also set up preclinical testing and phenotyping around any of these models. And you'll hear more about metabolic diseases today, but we also look at drug efficacy and have an entire suite in immuno-oncology as well. So again, as you can tell, I hope we're very excited by what we're doing. just in the times that I've been working with in mouse genetics, these technologies have all overlapped my education and training. So I couldn't be more excited now than I was 20 years ago. And in fact, what we really see, and one of the goals of Jim Pharmatech, is to be a key player in providing the technology, providing the animal models, and also the additional research to really have breakthrough therapeutics and contribute to breakthrough therapeutics in the next 20 years. Just a brief step back, just to point out, as I said, conditional knockouts. Essentially what Jim Pharmatech is doing is repeating most of the work that we did with the IMPC, but at this time, completely conditional ready state. So if you do not want a knockout from day one, but you want to be able to knock out genes in different tissues or at different times, we are now repeating, floxing all 22 to 23,000 genes that encode proteins in the mouse genome, and these will be available to our customers. And in fact, this project is nearly 1/3 complete. Keep in mind, our total capacity is over 5,000 per year, so we think that this project will be done in the next 2 to 3 years, if not sooner. Again, the flox mice don't do you much good unless you have something to use them with, and those would be Cree mice to induce the recombination. And we have over 200 mice available that have Cree-specific expression in different tissues and in different organs. And you can see the snapshot here of the wide variety of those that are available. And in fact, we already, with our collaborators and with customers, have seen nearly 200 publications using these conditional mice. No time to go into this today, but this will be the subject of another webinar in the future. We've humanized over 200 strains involved in immuno-oncology, particularly around checkpoints. And these are extremely useful models for checkpoint inhibitors, but also better understanding not just oncology, but also potentially autoimmune diseases. And again, just to sort of end, is that some of the breaking news for GPT, we've also developed an entire suite of humanized ACE2 mice. As you know, ACE2 is the target of the spike protein on the COVID-19 virus. And again, we've made these mice available to a number of researchers already worldwide. And we're very excited to report to you that a paper came out in Science just 4 days ago from a group that was developing small molecule inhibitors of the actual viral infection. So these antiviral activities was tested in transgenic models. And again, we're very happy to say that we contributed to that, as the mice were our mice that had the humanized ACE2 in the mouse genome. So again, just a very powerful example of where humanization is, and just to also let you know that these models are available. So I think with that brief but rapid introduction to Jim Pharmatech, I'm delighted to turn that over to Dr. Lee and allow her to tell you more about the NASH models and the liver humanized models. And again, if you have any questions, please feel free to enter those in the chat, and we'll answer them at the end of the talks.
Yeah, thanks, Mark. And also thanks to Pradeep for the kind words in your introduction. I'm going to share my screen. And oops. Okay. Right. So thanks everyone to join us today at this webinar. I'm very happy to talk with you today. And in the next 30 minutes or so, I will be introducing 2 of the models that we have been developing for NASH. And as you already can tell from the title of my talk, I'm already changed it. And near the end of the session, I'll be introducing some of the most recent developments that our R&D team has done in terms of, generating a liver humanized mouse model for some other liver conditions in the human population. So to begin with, liver, as we all know, it is the biggest solid organ and the biggest gland in the human body. It is a vital organ as well that carries out more than 500 different functions, including carbohydrate and fat metabolism, the synthesis of glycogen and cholesterol, the production of bile, which helps with digestion, Liver also detoxifies many chemicals that comes to the body and also metabolizes drugs that we take for our illnesses. And there are many kinds of liver disease as well. Fatty liver disease is one of the common ones, which is characterized by the pale color of the fat or the lipids that builds up in the liver, as illustrated in this cartoon. And the excessive alcohol consumption can cause fatty liver. At the same time, fatty liver can also develop in people who don't drink alcohol that much or drink very little. As a matter of fact, the non-alcoholic fatty liver disease is the most common chronic liver disorder in the United States. For the rest of the talk, I will use NAFLD to make the verbal delivery a little simpler and easier. In this slide, it illustrates how a healthy liver may progress from fatty liver to steatohepatitis with cancer or cirrhosis. A liver can remain fatty for a long time, decades, if not the entire human life. However, about 25% of NAFLD patients do progress to non-alcoholic steatohepatitis, which is NASH, the topic of today, after some form of liver insult. NASH is characterized by inflammation in addition to steatosis, which is the lipid deposition in the liver. The combination of the two leads to hepatocyte injury. About 25% of NASH patients actually develop fibrosis, cirrhosis, or even liver cancer in the long run. As you can see in this illustration, at the early stages of the disease, things are reversible with interventions such as diet, exercise, weight loss, or bariatric surgery, as illustrated by the blue arrows. However, if the diseased liver embarks on the journey as illustrated by the red arrows, the progress is not reversible anymore. Some patients may actually develop cirrhosis that require a liver transplant in order to extend the lifespan. So the incidence of NAFLD and NASH kept increase in the past 3 decades. As a result, NAFLD has become the most common cause of chronic liver disease worldwide, not just in the States. Given the data collected in 2018, which is cited here, the global prevalence of NAFLD is about 25.2%, and the NASH-associated cirrhosis and cancer has become the leading cause of liver transplantation worldwide. Parallel to this development, the research effort for NASH has been increasing also exponentially in the past decade, as shown by the number of publications in this figure. At the meantime, many leading pharmaceutical companies also put in their effort in developing drugs to alleviate this condition. In the same 2016 report, the global market for NASH has been estimated to be $0.8 billion in 2015 and projected to be $15 billion by 2025, which is only 10-year span. And this is a compound annual growth rate of 34%, which is largely driven by the introduction of several new premium priced products into the market. As of June 2020, just last year, Pharmaceutical Online reports that 54 clinical candidates are in development from 47 companies, and those 54 candidates are evaluating 29 different signaling pathways that related to NASH. So 2020 has brought a lot to us, whether we like it or we don't. It also brought to us a clearer perspective on this seemingly simple, but actually very complex disease. Numerous studies in the past 20 years have demonstrated that NAFLD is a hepatic manifestation of an underlying metabolic dysfunction. For example, obesity and type 2 diabetes are actually contributing to the development of NAFLD. It also accelerates the progress from NAFLD to NASH in almost all patients. And also has been reported that 80% of NASH patients are actually overweight or obese. 72% actually have dyslipidemia, and 44% actually have type 2 diabetes. So similar to metabolic syndrome, NAFLD and NASH are highly heterogeneous. As a result, a panel of expert researchers and clinicians reached a consensus and proposed a new terminology, a new name for this disease. Instead of NAFLD-NASH, they proposed MAFLD and MASH, which is metabolic associated fatty liver disease and metabolic associated steatohepatitis. So the panel believes that the new terminology better reflects the current knowledge of the disease and the pathogenesis of the disease. It also helps patient stratification for management during treatment, accelerates the translational path to new treatments. And in addition, it improves public initiative from a public health perspective. So with this updated definition of this disease, the diagnostic criteria also requires the evidence of hepatic steatosis accompanied by 3 additional features. First, overweight or obese. Second, type 2 diabetes. And third, lean or normal weight with the evidence of metabolic dysregulation. So with this in mind, from Zhiying Pharmatech as a model generator, supplier, and service provider for preclinical research, we strive to make sure that we provide the best mouse model for preclinical research, and we serve our customers' need in their effort to develop the best and novel drugs for human disease. So when we think about mouse models for human disease, we would like our model to reflect what the human disease is about as closely as possible. So with the new nomenclature for the disease and emphasis on the metabolic dysfunction aspect, we have made it clear in our R&D effort that our MASH model will adopt this view and this expert opinion. So before I introduce the 2 mouse models, I need to introduce this method to you. This is basically a method to evaluate NASH or MASH, as we call it. And this paper is published in 2005, before the introduction of the new terminology. So it still used NAFLD in the name of the method. But in a sense, this method is trying to use pathomorphological measurement to quantify the disease advancement of NASH. So for the sake of being simple and clear, I will use the same term, NAFLD activity score, NAS, for the rest of the talk. So in this publication, the researchers carried out measurements for steatosis, fibrosis, inflammation, liver cell injury, as well as some other parameters. And they quantified their measurements, which are basically morphological into numerical values. And all these values are summed up into one score, the NAFLD activity score, NAS, as shown on the right side. So the higher the NAS score, the higher the incidence of steatohepatitis. And the full score is 8. The maximum is 8. So now I'm gonna introduce some of the research that our R&D team has done when they are trying to put in the effort to develop new models. So they started with looking at and also doing comparative analysis of existing conventional NASH models. So as you see in this table, there are 6 models shown. The high-fat diet treated, the Western diet treated, mice with MCD, mice with CDA and high-fat diet, and also high-fat diet in combination with CCL4. And lastly, the STZ treated mice with high-fat diet treatment on top. So among those when they were evaluating these animals, we also, taking the new terminology in our practice, we evaluated in two sets of assessment. First one, as shown in the first 4 parameters, from obesity to hyperlipidemia. Those are the measurements for metabolic dysfunction. And then in the second set of assessments, we looked at liver morphological changes by looking at steatosis, inflammation, ballooning, and fibrosis. So as you can see right away, the highlighted area shows that none of these model is ideal. It's actually far from what we need. The 4 models on the right side, they don't have a good manifestation of metabolic dysfunction. And then the high-fat diet animal and also the Western diet treated animal, although they show good metabolic dysfunction as well as liver pathology, the treatment period of time required for the disease development is very long. So with this in mind, our R&D team has been working on developing these 2 new models for NASH, namely the BKS-DB NASH model and the ALMS1 knockout on the B6 background NASH model. So as you can tell from the name, they are a combination of genetic modification and Western diet treatment. So before I go into the details of some of the data that I would like to share with you, I'm going to give you an overview of how those 2 new models compare to the 6 conventional models that I've introduced to you in the previous slide. So as shown on the right side, these 2 new models, they have a good display of metabolic dysfunction, as illustrated by the red on the right side. And also, they only require a 4-week or 6-week Western diet treatment. So this will accelerate the research turnaround time a lot. So now I'm going to introduce the 2 models one after another one. This one shows you the metabolic panel that we assess with all the models that we are trying to work with. So 3 groups of animals are assessed in these experiments. The BKS wild type animal fed on the chow diet, the BKS DB animal fed on the chow diet, and also the BKS DB animal fed on the Western diet. As you can see, the DB animal fed on the Western diet, they have the highest body weight, the highest fasting glucose, the highest cholesterol and LDL. They also have the highest ALT and the AST, which are indicators for liver dysfunction. And next, we look at these animals for biomarkers of inflammation and fibrosis. We did this by mRNA expression, qPCR analysis. For inflammation, we look at TNF-alpha as well as MCP1. For fibrosis, we look at TIMP1, collagen-1, as well as TGF-beta. In all cases, the DB animal fed on the Western diet show a significant elevation of those inflammation and fibrosis biomarkers. And now, next, we come to the morphological analysis of these animals. As you can see right away, a 4-week long Western diet treatment made this animal to have this fatty liver phenotype readily observable. They have enlarged liver size. They also have the pale color, which is the lipid deposition in the cells. And to take a closer look by tissue sectioning and histological staining analysis, as you can see from the 3 columns of sectioning and images, these 3 group of animals, the last one on the right side, the DB animal on the Western diet, they show the highest amount of lipid accumulation, as shown by the oil red O staining in the lower panel. They also show the highest amount of steatosis, the ballooning of the cells in the liver, as well as inflammation as shown or pointed out by the black arrows on the top panel. And in some, we also quantified those morphological changes. As you can see in this table, these animals show a significant increase for steatosis and ballooning in the liver tissue. And they have an average NAS score of 5.8. The range is typically from 5 to 6. So we think they are really, exhibiting a NASH phenotype. With that in mind, I also want to point out to you, we try to keep transparency as much as possible with our customers for our research and our collaborators. So these animals, while they are showing a good NAS score overall, they do have a weak fibrosis phenotype as shown by the red line on the right figure on the right. So next, I'm going to introduce to you the ALMS1 knockout model. In our hands, we generate this model by CRISPR-Cas9 engineering, which is not surprising. So ALMS1 is a gene that is thought to be part of the centrosome and has a function in the organization of the microtubule. However, details on how this protein functions as such is actually unknown up to today. Spontaneous mutation has been identified in this gene among humans, and human patients who actually have this mutation, they have a condition which is called Alström syndrome. It's a rare genetic disease. Up to date, only about 900 patients has been reported worldwide. And these patients, they actually display a wide range of defects in many organs, starting from hearing, vision, heart, kidney, lungs, adipose tissue, liver, bladder, and skin. And a very interesting thing about this syndrome is that not all the patients display all the defects. And what is also found true about this syndrome is that many Alström patients actually display obesity and diabetes. So in this animal, what we have done is similar to the previous one. We fed them with a Western diet, and we measured their metabolic dysfunction in this experiment shown here. So again, 3 groups: the wild type on the chow diet, the knockout on the chow diet, and also the knockout on the Western diet. As you see, they have the knockout animal on the Western diet has the highest body weight, highest cholesterol, LDL, and highest ALT and AST. Looking at the morphology, after 6 weeks of Western diet treatment, their liver is large and pale. They have significant amount of steatosis, ballooning, as shown by the asterisks on the upper panel. And they have increased amount of inflammation, pointed out by the blue arrow. And then as a big difference in this model comparing to the DB on Western diet, this model actually show increased amount of fibrosis as shown in the lower panel. So overall, these animals, they display a average NAS score of 6, also in the range from 5 to 6 most of the time. In all 4 categories, this knockout animal on the Western diet, they are showing significant amount of all the parameters that has been measured by the morphology. So up to this point, I've introduced the 2 models to you. And in our research effort, we want to do experiment to demonstrate or to validate our model as a tool, or a very good tool to evaluate drug candidates for NASH. So in order to do that, we took 2 of the candidate drugs that is already in clinical trial, MGL3196 and the semaglutide, to test our animal and see how our animal respond to those 2 drug candidates. So as I said, Both of the two molecules, they are showing promising phase 2 data already. MGL3196 was originally designed for lowering blood cholesterol. It is a THR beta specific agonist. The semaglutide is originally designed for weight loss and for lowering blood glucose. It is a agonist for the GLP-1 receptor. So in our hands, we subjected these mice, these two models, to these two drugs separately and measured them as we have done previously shown to you, by the metabolic panel, by the inflammation, fibrosis, biomarkers, as well as the morphological assessment. For the interest of time, I'm going to show you data from the morphology assessment because I think that's the most indicative, illustrating how the liver in the animal is responding to the treatment of those two drugs. So showing you here is the BKSDB animal fed with a Western diet and also treated with the MGL3196. As you can see, or if you focus on the middle and the right panels, these animals that are treated with the vehicle or the drug, and looking at the amount of lipid deposition by the red staining in the lower panel, and also the ballooning, the steatosis, and inflammation by those arrowheads, you can see there's a significant reduction on all parameters that we are looking for in these animals. And then to sum up, using the method of the NASH score in all the three categories that measured steatosis, ballooning, inflammation, the drug is able to reduce these phenotype significantly. So our animals are responding quite effectively to the drug treatment. Similarly, we subject those BKSDB NASH animals to the semaglutide treatment. Also, similar to the display in the previous slide, if you focus on the middle and the right, the treatment with the drug reduced steatosis, reduced inflammation, as well as the lipid deposition in the liver sections. And then to sum up, as a a numerical score, the NASH score, the lowering of the steatosis, ballooning, and inflammation is readily is observable from the data. So up to this point, I've shown you data of our model responding to those drugs. I will also want to show you, with our experiences, with our hands-on work with those animals, knowing everything about them, the study design we have in terms of timeline is illustrated here. So before we start, we're going to do a QC step in order to eliminate any outlier animals, which is due to the biological variations among mice just naturally occurring. That's our first QC step. We'll measure the mice for ALT, which indicates liver dysfunction, for fasting blood glucose, as well as body weight. And then after we get animals to be a coherent cohort for the study, we randomize them into different treatment groups. And in each group, we have about 8 to 10 animals. And next, we'll subject them to the high-fat diet, or the Western diet treatment for 4 weeks. In the meantime, we'll start dosing the animals with a testing article for the research. We can do dosing through oral intake or intraperitoneal or a sub-Q dosing pathways. We can measure their body weight, their food intake, their fasting glucose, HbA1c, and also a panel of blood chemistry. These are mostly driven by customer request. After 4 weeks of the diet treatment as well as the dosing, the research where the experiments come to an end. At the termination point, we're going to subject the animals to a comprehensive panel of analysis, which include blood, urine, tissue collection. The blood chemistry is the full panel of everything that is tested, a histological analysis of the tissue, as well as gene and protein expression like qPCR or Western. So in conclusion, for this part of the discussion, I've introduced to you these two models, the BKSDB as well as the ALMS1 knockout on the B6 background. These two genetic mutations actually subject the animal to an early onset metabolic dysfunction. And at the same time, when they are fed on a Western diet, the diet treatment basically accelerates the liver morphological worsening in the animal, as shown by the steatosis and the other parameters that has been assessed in the study. So in our hands, and given the data, we believe both models are suitable for efficacy evaluation of drug candidates for NASH. And with the last slide, I just want to introduce to you, today is our second webinar of the year, starting from 2021. We're going to have series of webinars planned for our audience who has the interest, and I have listed out the different topics for March until July for this year. We're going to talk about ACE2 animals, as Mark alluded to, for the COVID-19 research, the immune checkpoint portfolio. We have a lot of animals that we can offer and services. The immune deficient animals is a great toolbox for people looking into all aspects of immuno-oncology, and also germ-free mice for microbiome research. And finally, we will have discussions on the GPT's gene editing platform. So with that, I'll say thank you for your attention and time. And as I said, we encourage you to contact us if you have questions on the models, if you have questions on the research we have done, or if you want to collaborate with us anytime by email, by phone, and also look on our website. There are a lot of information that you can browse through. So with that, I'll hand it back over to Pradeep.
Thank you very much, Zhiying. Thank you very much, Mark. What a fantastic two talks. I told you, you guys are in for a treat. So all the attendees, you can have questions and answers, and ask your questions, please, at the bottom in the Q&A chat box. Actually, we had a great question during the talk itself by Vincent Thomas. What a name, Vincent Thomas. And he said, when do you expect to be able to ship mice from the USA facilities? Thanks, great presentation. How come you didn't say great moderation? But anyway, Vincent, I still like you. I still like you, Vincent. But Mark kindly answered that question. So I'm going to say that question out again, so Mark can answer that to everyone attending. So Mark, how would you answer that question? When are you able to ship mice?
Depends on when the orders come in, but we are operational and ready to go. We are very excited. As our press release said, we've secured animal facilities on the East Coast. We have the capability as well on the West Coast to take in frozen embryo and sperm from China, where we can reanimate them and then provide them to customers. And we also could take in live mice from China, or take the reanimated mice and expand them and make shipments to customers. So again, we'd work with any orders and work on the timing, but we are ready to go. And thanks, Winston, for that question.
Yeah, a great question, and enlightened everyone. Winston, your name actually inspired me. Being British, I like Churchill, and when the great man was approached once, he was asked, what do you think about perfection? And Churchill said, you have to improve, you have to change, he said, but to be perfect, you have to change often. So that's what we're trying to do in Gem Pharmatech. We're taking these models and we're changing them, we're kind of adapting them, and you can see how Zhiying said that you took a 16 to 30 week model, and we now have it into 6 weeks. So kind of I did my PhD and I had 30-week OA models. So Zhiying, I wish I took metabolism, and I'm using these new models. So at Gem Pharmatech, we're always changing, trying to be efficient, trying to customize things. And everyone that attends, we want to tell you that we're more than open. We can't be perfect, but we're more than open to take your custom orders and serve your needs and tell us your requirements. We're kind of a one-stop shop, and we do a lot for all our customers. Having said that, we do have some more questions here, Zhiying. Let me ask you here. Someone asked, how do you measure lipid content in the mice?
Well, lipid content is, actually a very simple measurement. We take the blood from the tail vein of the mice. Other people can take from other parts of the body of the mouse, and then we subject the collection to centrifugation, basically to separate out the serum, and then you have kits that you can use to measure the lipids in the serum.
Okay, great. And we actually got a very interesting question. And it's a well thought out one. Where do the hepatocytes of your liver humanized mice come from?
That's actually a very great question. Although, I mentioned we take the hepatocytes from healthy human subjects. It is true, we take from human subjects, and they are primary cultured cells from the human. They are not, passaged, cultured human cells. So there's a big difference between these two type of cells. So great that people ask this question to make sure.
Good. Thanks for that. And we'll probably end on this last question. Are these models on sale?
Yes.
That's a very sweet and short answer. It's a timely answer as well. So I want to thank Mark and Zhiying again. Great presentations. And please, everyone, just contact us on globalservice@gempharmatech.com if you have any questions related to any mice projects or even the NASH projects as we discussed today. So thank you, everyone. Good night from London. Good morning wherever you are. Good afternoon. So hope to see you guys soon in other webinars as well. Thank you. Bye.
Thank you all.
Thank you. Bye-bye.
Thanks for joining.